we're going to look at how do plants interact with pathogens. This is a very important chapter because it really goes in as to what's happening with plants and what plants are doing when the pathogen comes in and attacks them. We're going to break this down into several different parts. There's going to be a part on ecological interactions, which are really what happens between the plant and the pathogen as far as general concepts outside. Where do pathogens come from? We're going to look at the physiological interactions, which are what is going on physiologically between the pathogen and the host. We're going to look at the genetic interactions, which again goes back to the genetic part of what's happening between the plant and the host, how the plant is favored in the interaction here. When we look at these, we're going to deal with all of these different parts. We're going to start out with the ecological interactions. Ecological interactions. We're going to look at these three basic parts. How do plants survive in the absence of the host? How do pathogens find plants? How do pathogens penetrate plants? How do pathogens survive in the absence of the host? When we go back to the disease triangle, we are constantly trying to figure out about the favorable environment. What happens if the environment's favorable for the pathogen but is not favorable for the host? The host isn't growing. That's not a good scenario for the pathogen as well. How do they survive in the absence of the host? There are resting structures of pathogens, which include things like oospores and zygosporangia, which have the ability to remain in the environment for prolonged periods of time where they are not actively growing. Then once you get a stimulation from the host plant, they will start to grow. So here we've got a picture of an oospore. We've got sexual structures, which can be cleistothecia, perithecia, apothecia. They will remain viable during the time when things are not favorable for the pathogen. Sometimes we call this overwintering, going from one year to the next. We have what we call chlamydospores. These are thick-walled spores. In this particular case, they are asexual spores. These will remain in the environment waiting for proper conditions before they start to grow again. They are very common. We have things like nematode cysts filled with eggs. The nematode cyst we talked about will go from one year to the next, overwintering without germination of the eggs. Then in next year, when the conditions spring up the way they should, all of a sudden you get hatching of the eggs, you get the release of the new nematodes. We've got perennial cankers. This is on pine tree. This lives from year to year, and in the springtime when everything comes up, right, boom, it comes out. It just starts moving on right from there. We have got what we call sclerotia. Sclerotia are hardened structures of the fungal pathogen that have the ability to pass from one year to the next without being destroyed. Sometimes they can go for many years without being damaged. And when the climatic conditions are correct, they will come out. This is Sclerotium rolfsii. This actually came up in the garden. We had not had this particular pathogen there, which means it came in somehow on its own, or it had been there for a prolonged period of time. You can see the sclerotia start out kind of whitish, then they get darker and darker browns, and eventually they become these hard little structures that can be transmitted rather easily with the pathogen, or they can resist in the soil until they get new host plant coming in on them. Another strategy, there is survival in perennial plants and seeds. One of the concepts we talk about is alternate hosts. Theoretically, the alternate host is you have that host which you're trying to grow. You also have another host that may not be what you're trying to grow, but also serves on that pathogen. This is very common on rust fungi, because with the rust fungi, we have these different stages. This shows you fusiform rust on pine tree. You can see it's got all of these symptoms. Theoretically, you'd want to remove any tree that is problematic with this. Its alternate host is live oak, which is an absolutely magnificent plant that we have in the same area. The question is, which is the more desirable host? If you're looking at it from an economic point of view, it may be that the pine tree is. If you're looking at it from a point of view of landscaping and everything, without a doubt, the live oak is. You've got these two hosts. The iridial and the teleal phase occur on pine. The pycnial and acial stage occur on live oak. Chances are you'd never even notice the disease on live oak. We've got those two different parts. You can have perennial things occurring on plants. This shows you anthracnose of bean, and you can see the rather nasty necrotic lesions that you are getting on the outside of the plant When you harvest the seeds, you can see how seeds in here become very colored. That is due to this particular fungus on these plants. It is a way that it can pass from one year to the next on infected seed. If you aren't careful about making sure that your seed is properly treated, you're going to have major problems. This is Diaporthy phaseolorum on soybean. You can see the soybeans don't look very good. When you plant these soybeans, what happens is instead of germinating, the mycelium of the fungus comes out and it contaminates everything. The picture down below is actually what the fungal structure looks like when it forms on the plant. They're surviving in the absence of growing host tissue. We talked about Anguina triticide. You've got your seeds up at the top. On the left-hand side, those are normal seed. On the right-hand side, they are infected. You take the infected seed, you put them in water, you crush them a little bit, you get these thousands of nematodes crawling out of them. You get transmission along with the host part. This gets to be very problematic. It survives from one year to the next in the seed. Since we understand this, in the United States, Anguina triticide has just about been eradicated. It's very easy to pick out the problematic seeds, especially with modern technology. How do pathogens survive in the absence of the host plant? It can survive in a vector. This is a picture of what we call ambrosia beetles. The little beetles basically come out, they burrow in, mama lays the egg. As mama lays the egg area gets contaminated by the fungi that mama carries on her outside. The little guys that feed on the wood, they also are in close contact with the pathogen. As they do that, the pathogen gets on them. When they pupate, mature, and come out as the adult organism, it's stuck on the outside of them. The adults go in, lay the eggs, and as they do that, they contaminate the pathogen. This survives in a vector from one point to another. When we talked about viruses, we talked about persistent type viruses, where it can remain on the stylet. We do see things like this as well. So survival in vectors is very important. Survival on plant debris. He's going out there, he's plowing the field. Did he wash his implement in between the time he plowed his last field and he came out over here and made sure that he's not taking any soil with him? I doubt it. When this happens, you have a very good way of transmitting pathogens. It becomes very problematic. If you look at this corn stalk on the outside of the corn stalk, you've got all of these little spots which are pathogens. Their ability to survive from one year to the next depends on how well they can compete with other organisms that are going to be trying to get the same substrate during the time that it is overwintering. There is a lot of survival. We have survival in the soil. We talk about soil inhabitants. These are things that are naturally in the soil that can survive on the soil on their own. And then when the host comes in, boom, they're going into it. A good example of a soil inhabitant would be Rhizoctonia solani. It occurs everywhere in the soil and it is very problematic. We have what we call a soil invader. These are not normally found in the soil. They can come in and they can try and survive in the soil once they've been incorporated in the crop residue. If they can survive from one year to the next, then they will create disease. Their challenge is they have to be competitive against all of the other microorganisms that you find in the soil. It says here they can survive as epiphytes. What does that mean? They're on the outside of the plant, not necessarily on the inside. When we look at survival, we look at a lot of different mechanisms on how these things can survive in the environment without necessarily having the host present.